Dear all, hereby I open this academic ceremony in which Matthew Davies will defend the academic thesis, Colonization of the Gut Microbiome by Escherichia coli during international travel. May I invite you to present a summary of your findings, of your studies, and the conclusions of your thesis.
Thank you. Dear Pro-Rector, dear committee members, dear supervisors, friends and family, both here in person and online, thank you for coming to my PhD defence here in Maastricht. Over the next 15 minutes, I will present some of the work that I carried out during my PhD, which is titled The Colonisation of the Gut Microbiome by Escherichia coli during international travel. I will start by introducing some of the fundamental concepts behind my field of research, namely antimicrobial resistance, the gut microbiome, and Escherichia coli, and how the work that I carried out during my PhD links these three topics together. So firstly, what is antimicrobial resistance, or AMR? AMR is when bacteria gain the ability to protect themselves against the use of antibiotics. This is then a problem because antibiotics no longer work to treat infections that might be caused by these AMR bacteria. This is a problem around the world, especially in low and middle income countries, and is such a significant problem that in the next 25 years it's predicted that 10 million people per year will die directly due to AMR infections. This also doesn't even consider the total number of infections from AMR bacteria, any chronic illnesses and permanent disabilities that might be caused by these infections, and also doesn't consider the economic burden from the healthcare costs associated with treating these infections. The bacteria, which is the main focus of my work, was Escherichia coli, or E. coli. This is mostly known to be associated with food poisoning and UTI infections, but it is a bacteria that does just live normally within the guts of people and animals, and also the environment, without causing infection. However, E. coli is also really associated with AMR, and is actually the world leader, or one of the world leaders, in AMR infections and the associated deaths. It's particularly a problem within E. coli due to a concept known as multi-drug resistance, where E. coli has the ability to protect itself against various different types of antibiotics that we may use to treat infections caused by E. coli. This by itself isn't always like a death sentence. You can just be a carrier of this AMR E. coli without it causing infection. And this is something that is then becoming more relevant due to an increase in globalization that we are seeing as time goes on. Tourism is such a big part of a lot of countries' different economies. So the transmission of this AMR E. coli between countries is becoming more of a problem. You do see here that the rates of carriage of AMR E. coli without necessarily causing infection does vary across the globe. And specifically in Europe, we do have the lowest rates, especially Western Europe, of approximately around 6% of people are carriers. However, when you then start to look at places like Southeast Asia, this can increase to as high as, some studies finding it, as high as 60% of people are carriers of AMR E. coli. Like I briefly mentioned, this is then a problem when you start including international travel. And you see in a study that was the precursor study to the work that I did in my PhD, they found that international travellers going to these countries, especially like places like India, for example, had nearly 84% of these travellers ended up picking up this AMR E. coli during their travels. We know that the risk factors associated with picking up this E. coli is the place that you go to, because some countries have higher rates of endemic carriage. If you develop traveller's diarrhea during travel, if you use antibiotics, and also if you have irritable bowel syndrome or a disease. The interesting thing about three of these factors is that they are associated to changes in the gut microbiome. But what is the gut microbiome? So the gut microbiome can be defined as the ecosystem in which all of the microorganisms and the host interact with each other. A healthy gut microbiome is very important and is useful as it helps to digest nutrients, ones that we might not be able to digest by ourselves. We need the microbes to do it for us, but also reducing inflammation and preventing stuff like diarrhea. And most relevant to my research is the protection against potentially invading harmful bacteria. How this is achieved is with a concept known as its microbiota diversity. So here you can see that if you have a high diversity, so many different species of bacteria that occupy all of the different niches, all of the different areas, and use up all of the nutrients within the gut microbiome, or within the gut, is innately protected against invading, and in this case, invading E. coli. However, a low diversity, which is often associated with stuff like diarrhea, is then a lot more susceptible to being invaded by harmful bacteria like E. coli. So then this links to the work that I carried out during my PhD. So in the study design of this is we have fecal samples from before travel, travellers that went to 4 different regions around the world, which is North and East Africa, South and Southeast Asia, and then took fecal samples again immediately upon return. A bit over half of these people ended up picking up AMR E. coli, and a bit under half of these people did develop traveller's diarrhea during travel. And we do have a low usage rate of antibiotics that is not then therefore confounding our results. And my first main question that I wanted to answer is to discover how the gut microbiome might differ between people that do and do not pick up resistant E. coli or AMR E. coli during travel. The way I did this analysis is with something known as bioinformatics, which, to summarize, is working with the sequencing of DNA. So to sequence DNA, you have to take its genome and break it up into loads of really small pieces. And it's kind of like doing a jigsaw puzzle. So you have all of these individual pieces, and then it's my job to put these pieces back together. However, because I did this on a fecal sample, this contains many different species and the genomes of many different species of bacteria. So it's kind of like having thousands of jigsaw puzzles, throwing all of the pieces together, and then it was my job to separate all of these out and build these individually. And it's these which I can then identify as the species of bacteria. And then from therefore can start to determine stuff like the diversity within the microbiome per person. And these are some of my results. I do see that the diversity before travel, so from the initial fecal samples, do not differ between people that do and do not go on to pick up AMR E. coli during travel. Also, when I compare the diversities from people before travel and after travel, I then also see no difference between people that have picked up AMR E. coli and those who have not. Meaning that the diversity of the gut microbiome is then therefore not linked to whether you're susceptible or not to picking up resistant E. coli during travel. However, what I did discover is that instead of looking at the diversity, it's actually the presence of specific species of bacteria that is then potentially protecting you against picking up AMR E. coli. So this example, I discovered it was Citrobacter fungi was one of these bacteria. So I wanted to test this. I wanted to see how Citrobacter may be able to compete with the invading E. coli. So I did a competition analysis. I added equal parts of Citrobacter and E. coli into a nutrient broth and allowed it to grow for 8 hours. And then this growth of this bacteria you can then put onto a Petri dish and you can count the numbers of these bacteria. What I expected if Citrobacter could outcompete E. coli was that when I then count the numbers, I see a lot more Citrobacter than I do E. coli. However, what I ended up seeing was not the case. So I know that in this experiment that Citrobacter, if it is outcompeting E. coli, is not doing it in a direct manner, so it's not killing the E. coli. As a slight alternative, I also noticed that when I compare the amount of E. coli that people have, the quantity before travel and after travel, even in the people that we know for definite picked up AMR E. coli, I don't see an increase in the total amount of E. coli that they acquired during travel. So I started to speculate that there's something to do with the E. coli diversity. So we know, as we've seen with the COVID pandemic, there's this concept known as variants. It's the same virus, but a different variant can come along and cause different levels of infections, and it might be slightly worse. And it's a similar concept with E. coli. We have many different strains of E. coli. I then looked at these strains, these specifics in these people, and saw that after travel, only those that contained a specific strain of E. coli before travel were always then replaced by E. coli they picked up during travel, whereas it was not the case for other strains. So it's suggesting that the specific strains of E. coli are then leading to you to be more susceptible to picking up the invading resistant E. coli. And with that, I'd like to conclude some of the work that I've done during my PhD. The main findings I found was that the microbiome diversity as its entirety, so the microbiome, when you really look at the whole thing, is not the leading cause of why we pick up resistant AMR E. coli. And I think the next step for exploring this is to then, instead of looking at just the species, look at all the molecules that are present within the microbiome, all of the molecules that the species are producing, and maybe this could point to certain patterns of molecules within the microbiome that could be protective instead of the species themselves. I discovered specific species that might be protecting you against resisting E. coli from colonizing you. And the next experiment for this would be to test this in an environment that better resembles the gut microbiome. So the gut microbiome innately is kind of hostile for bacteria. There's a lot of competition they have to do with all the other species of bacteria there and against the host. And they have to fight a lot harder for stuff like nutrients because they're available at a lower rate. So maybe Citrobacter, for example, the protective species, are better at handling the stress than the invading E. coli, and that could explain why they are protective. I also discovered that it is specific strains of E. coli that might lead you to being susceptible. So it'll be interesting to determine how these strains at a very, very specific level are interacting with each other within the gut microbiome, and whether this could explain that some strains of E. coli are less able to kill the invading E. coli, for example. And with that, I'd like to end my presentation. Thank you for your attention. Bedankt voor uw aandacht. And I give the word back to the Prorector.
Thank you very much, Mr. Candidate, for your crystal clear presentation and also your future perspectives on travelers' diarrhea. Your position will be opened by Professor Huber. He's a chair in social medicine, in particular infectious disease control at Maastricht University. Professor Huber.
Yeah, thank you. Dear candidate, congratulations with this very nice thesis, and I also want to congratulate the supervising team, of course. I've read the thesis with interest, and I'm really happy to discuss with you some questions. But I'm not a microbiologist. I'm from the public health, so my questions will be a bit maybe out of the box. And I want to talk about ESBL, Enterobacteriaceae. It's a really interesting but also quite puzzling problem. And you refer to your low and middle income countries, of course, when talking about acquiring ESBL as part of traveler's diarrhea. But actually we don't have to go that far to find quite impressive differences in ESBL carriage in healthy people. Last time when I had to do with ESBL was a few years ago when we studied ESBL in just around the corner actually in Belgium. We did a cross-border research project in Belgium and the Netherlands, and we performed a study in fecal samples in long-term care facilities, so elderly residents, and also in daycare centers for children, so babies, to look at the public health impact actually of ESBL. And what we found was that in Belgium, in 700 residents, the percentage of ESBL was 16%, and in the Netherlands, in these residents, so 600 residents, it was only 6%. Okay. And when we looked at the daycare centers for children in babies, we found also in Belgium 16% and 6% in the Netherlands, so a 10% difference. So the outcome was puzzling in two ways, actually. Because we looked at people from the start of life and also somewhat at the end of life in that sense. And they were being the same within the country. As you explained in your introduction, the gut microbiome develops actually from early life. So it's interesting that it's in both stages of life in a similar way. And on the other hand, two countries, a lot of similarities, of course, and next to each other, also a large difference. So I'm really happy that I'm here now with an expert in this topic, that's you. So could you help me explain, on one hand, the similarities, but also the differences?
Mm-hmm. Highly esteemed opponent, thank you for your good question. Yeah, that's a very interesting and puzzling situation between these two countries. I'm not really that intuitive when it comes to Belgium as much, and the like stuff like the rates of antibiotic usage within Belgium compared to the Netherlands. I have heard that in the Netherlands, antibiotic usage is relatively controlled, relatively low. So I can speculate that if there's a difference there, the use of antibiotics more frequently during life, at least. So this might explain more people in the care facilities. Is then you're kind of having an impact on the structure of the gut microbiome consistently through life if you use antibiotics more frequently, which then maybe constantly or repeatedly perturbing the gut microbiome structure, and then allowing stuff like ESBL E. coli to live within the gut, or you can pick it up more frequently. And then, once it's in your microbiome, the long-term carriage of the E. coli that we have here could be more specialized at long-term colonization of the gut microbiome. So if these are then the ESBL E. coli's, all the plasmid itself just remains within the gut microbiome. Could be why, at least in the care facilities, we're then seeing higher rates in Belgium.
And the funny thing was that, of course, we also looked at differences, and we looked, for instance, there were some slight predictors change in travel, for instance. Mm-hmm. That was something that was, but and also with linking to cattle or farms, and even within the antibiotics there was a slight difference, but all couldn't really explain this difference. Mm-hmm. So maybe it wasn't in this setting that the antibiotic was the driver, but of course it could be the case in general practice, for instance. So that's a bit what you were looking at as well. In general, there is more antibiotic use, and that's something we know from Belgium that is probably the case. Mm-hmm. And do you think this is a problem, that we have these numbers? Is it a public health problem where we should do something about it, and should we expect increases now when there's so much traveling going around?
Yeah, I definitely think it's something that we should be keeping an eye on, of course, because if a neighbouring country has significantly higher rates of carriage, then it could be a problem for the Netherlands.
Because there's probably quite some mobility between Belgium and the Netherlands and vice versa. Kind of traveling like that. Is that something we should do something about?
I think
close borders.
Yeah, it's touching on a quite tricky topic on how do you actually control stuff like movement of people, because you can't really put anything in place to stop movement of people between Belgium and the Netherlands. I do suppose if we're seeing an increase in Belgium and then that's in line with an increase in the Netherlands, then it could be a reason, we know that's the source of this, and then if you know, I don't know, if like someone's been to a hospital in Belgium and you can ask this when they come into a hospital or a care home in the Netherlands, considering stuff like Belgium could then raise a red flag to be like, okay, this is a place that might be, they might be more likely to be containing or carrying E. coli, and then make informed decisions at more of a specific level. But I do think at a national level, yeah, obviously, there's can't put things in place to stop people.
But do you envision that we are heading towards India, like that kind of percentage is also in Europe?
No, because I think the practices between different countries are just so drastically different. So just the rates of stuff like antibiotic usage and in agriculture within the accessibility of antibiotics over the counter is just innately different. And then we have a well-established infrastructure in place to be able to counter some of these things. So I don't ever see it increasing to the point of the same level necessarily as India, or 83% picking up for carriage. But there are trends over time of ESBLs are increasing over time, so it is becoming more and more of a problem. Yeah.
Okay, thank you very much for your question and answer. And I give the word back to the prorector.
The opposition, thank you very much. The opposition will be continued online with Professor Jagigan. She is Professor of Microbiology and Infection at the University of Birmingham.
Hello, and thank you very much to the candidates for an excellent, clear summary of their thesis. It's very enjoyable. And so I would like to start off the discussion by asking or by exploring the topic of colonization resistance. So I was hoping that you could begin this discussion by telling us what you understand by the term colonization. This is something that your thesis has given us some nice insights into, so it'd be interesting to hear what you understand by the term colonization resistance.
Yeah, to me, colonization resistance of the gut microbiome, I really define that or interpret that as like a state of the gut microbiome that then is just innately protected, obviously, against bacteria that want to thrive within that environment. So stuff like with E. coli to be colonized with really thrives in an inflamed environment. For example, not having inflammation is then innately colonization resistance to bacteria that might want to take advantage of an inflamed environment. But then there's also, it depends at what level you go down to, because you can have colonization resistance at the concept of like within the gut epithelial cells, the fact they've got like a mucus layer, and then Akkermansia, for one species, lives in this area, E. coli can't come in and then physically occupy that one very specific niche. So that's also then colonization resistance. And then other factors, just stuff like some of the metabolites, like short-chain fatty acids, again, is another aspect of healthy gut epithelial cells because of the production of stuff like short-chain fatty acids. Means that there's a functioning host immune system that can fight off anything that it detects as foreign that's potentially causing a problem. So it's kind of a nuanced answer with loads of different avenues and aspects to it. But yeah, that's pretty much kind of what I would take from colonization resistance.
Okay, thank you for that nice summary. In your thesis and in your presentation, you discussed the idea that there could be molecules produced by the gut microbiota that might be contributing to colonization resistance. So could you tell us a little bit more about this and what you believe is happening and what you believe the messages could be for future prevention of colonization by AMR-resistant bacteria, for example?
Yeah, so like I touched on that briefly with the concept of short-chain fatty acids. These are when you get dietary fibers, bacteria then can metabolize these fibers for nutrients and then produce these fatty acids, which then have a lot of beneficial effects on the gut microbiome. So when I saw stuff like the increase of Bacteroideaceae within the gut microbiome of people that did not pick up ESBL E. coli, Bacteroideaceae are big producers of short-chain fatty acids. So that could be a reason why they are then therefore kind of protective, and could be one of the molecules that is leading to a quote-unquote healthy gut. I suppose there are some other concepts as well of like the host-derived bile salts. These are then also frequently used by healthy bacteria, and then molecules that like that can help bacteria that aren't causing harm because they're not causing inflammation, they're just kind of coexisting within us. If they're able to promoting those types of bacteria, then it's another molecule that we could then sit there and say is contributing to a effective colonization resistance.
Okay, thank you. Exploring that in a little bit more detail and thinking about the gut microbiota and its role in colonization resistance, do you feel that molecules such as bacteriocins or toxins secreted by type 6 secretion systems could also be important in this niche?
I think the fact that I then see a displacement of E. coli strains within the microbiome, these pre-travel strains of E. coli might be species that aren't producing the correct bacteriocins or microcins, at least with Enterobacteriaceae, to kill the invading E. coli. Same with the Citrobacter, the fact that Citrobacter fundii comes out as potentially beneficial. Again, is within the same family as Enterobacteriaceae. If this is then also producing microcins that are specific microcins that are then beneficial. So I do think that's why I touched on it briefly when I looking at the metabolome, if these would start to be pulled out, these microcins, or looking at very specifics, really do like a microcin-focused study of different strains of E. coli or different strains of Enterobacteriaceae, and if that's the way that these are outcompeting with each other. However, I didn't see that, at least in the competition analysis that I carried out. So at least with the strain of Citrobacter I used in that analysis, isn't producing a microcin that's effective against ESBL E. coli within a highly nutrient population. Maybe with under stress, then they triggered the production of these, and that could explain potentially why.
Yeah, it's a really interesting point about the stressful environment and how the conditions within the gut, for example, could be influencing production of these molecules or increasing the potential for competition. Have you any thoughts for us or any ideas for us about how you could model this outside of a person's gut? So how you could model this in a lab environment, perhaps?
So I do know there's a model, the TIM2 model. I think here in Venlo, I think, where it's a lab gut microbiome environment essentially, where you can have a microbiome present that may contain species of interest, like Citrobacter fundii, or these specific strains of E. coli, and then we can determine when you spike in invading E. coli, which ones are then let it grow, and use this model that like tries to resemble the gut and all of the digestion and stuff like that that happens within the gut. If you then run this model, might then be able to pinpoint which species or which strains are still present after having it grow for a specific amount of time. So yeah, I think the TIM2 model would be where I would focus this next.
Okay, fantastic. Thank you very much for the interesting discussion, and that concludes all of my questions.
University appreciates that you were online present from Birmingham. Thank you very much. The opposition will be continued by Professor van Zorgen on site, and she is like the previous opponents, member of your assessment committee who already assessed your thesis. She is a professor of translational microbiology at Amsterdam UMC.
Thank you very much. Dear candidate, first of all, I would like to congratulate you on finishing this work, and I would also like to include your supervisory team in my congratulations. It reads really well, It's nicely laid out, clear as your presentation, and I learned a lot from it. I would like to start with you on chapter 2, where you immediately describe in the results section that actually you recovered 148 ESBL strains from 103 travelers. So that means that there were actually travelers that more than one strain was recovered from the sample. And I wondered if you explored that a little further. If there was any information in that, like, were there people that had 4, and what were they? Were they all E. coli's, or was there maybe an antagonism between different ESBL carrying species? Could you maybe say a little bit about that?
Highly esteemed opponent, thank you for your kind words and your good question. Yeah, so I have read before that some studies have seen that most people tend to have a dominant E. coli, for example, but then the presence of rarer species may exist within the gut microbiome below detection, or just multiple species do exist within the gut microbiome. Within the bounds of the metagenomic data I had, I couldn't detect this myself. So I did try to build metagenome assembled genomes, and per person only ever saw one strain of E. coli in each person. But that's not to say that they obviously don't have multiple different strains.
But these are cultured strains, right? Are they
From
not?
the combat study, yes.
Yes.
Yeah.
So how is there the association between having multiple species present in a single person?
I haven't explored
That part,
whether these people with multiple and maybe then always have ESBLs or not. I can imagine that the chances are that you've picked up ESBL E. coli during travel, and then it's just coexisting with your resident E. coli, and that's why you may then be able to detect multiple different species or multiple different ESBLs.
Right.
Whether that is you've picked it up during travel and then horizontal gene transfer has occurred of the plasmid, because the ESBL machinery exists on a plasmid, has this then transferred to other species within the same family, and that's why you can detect multiple. Some of these travellers did go abroad for like 3 weeks, for example. Yeah. I think a nice way of really answering your question with this is to do, if you had daily sampling during travel, you could then see whether maybe day 1 they pick up an ESBL E. coli, and then you can see the expansion of maybe the plasmid or other ESBL E. coli's of different strains within the gut microbiome during travel. And then obviously when we took a sample post-travel, we then detect all of them.
Yeah. That would be a very interesting thing to actually study. I was also intrigued by your finding on the possible protection from acquiring ESBL E. coli by Citrobacter freundii. But interestingly enough, you describe again in this first culturing result Citrobacter freundii as one of the ESBL containing species. So then I started to wonder, is this species now part of the problem or part of the solution?
Yeah.
Or does that depend?
I think it's a nuance. Like, it depends, really. I think there's a couple of aspects with this. Is the way that I carried out the analysis by doing the co-abundance gene clustering. So I've just got a bunch of genes that exist at the same ratios amongst different people, and that's how I can then go, that's probably belonging to a single species because they exist in the same proportions. I've then identified those genes as belonging to Citrobacter freundii. I do think there's a slight bias towards the databases are so focused on Citrobacter freundii compared to other Citrobacter species, like Citrobacter amylonaticus, which I explored within chapter 3. So I can't, it is identified as Citrobacter freundii, but these might be core genes, for example, within this cluster, which then most likely will always be annotated as Citrobacter freundii, because it might be a core gene within the Citrobacter genus. So I think from that aspect, I wouldn't be surprised if these were another species of Citrobacter. But then saying that as well, within the bounds of the study, the rates of Citrobacter being the carriers of ESBL coli, or that picked up after travel, are significantly lower. Like it's predominantly E. coli.
Yeah, no,
I think
that was very clear.
Yeah, it's like a few, maybe Klebsiella, and then very much like singletons with Citrobacter.
That could maybe be an in-host horizontal gene transfer, for
Potentially,
example?
yeah. It could also exist as with the carriers within
Citrobacter.
the Citrobacter within that country. Or within host transmission as well. Yeah, for definite.
So do we know anything about the population structure of Citrobacter freundii? Or is there a lot of different clades that maybe could have different functional
I have
effects?
not looked into clades of Citrobacter freundii. I do see quite a lot of species of Citrobacter.
Right.
It's within chapter 3. There's a phylogenetic tree of the genus of Citrobacter, pretty much. So there's quite a lot of species, and I do know that these do occupy different environments. Like some of them are more associated with just environmental, some of them are more with animals. Within Citrobacter freundii itself, I have not seen any clades, for example.
Right. So moving on to chapter 3, where you explored maybe the functional interaction between Citrobacter and ESBL E. coli a bit more. As you mentioned already in the presentation, actually E. coli outgrows Citrobacter, which was kind of unexpected. But you also said during the presentation, is therefore not producing maybe a direct antibacterial component. You do mention in the discussion that some Citrobacters, I'm not sure which one exactly, are able to produce hydrogen peroxides, which then could potentially be toxic to E. coli, probably not differential between ESBL E. coli and E. coli.
Yeah.
But I wondered whether, since you knew about this probably beforehand, did you check whether the culture conditions that you used to do these competition experiments actually allowed production of hydrogen peroxide? Is that a factor in the assay?
It was not, no. I really wanted to do the initial, like the foundation step of that type of analysis was really if they've just got plenty of nutrients and they're not in any stress. I didn't consider stuff like hydrogen peroxide and whether they are producing that. I did just want to see at that point, are they, when there's fully nutrients, are they able to outcompete or not? I do think the next steps would be really trying to limit the nutrients that are available and then consider stuff like, is this then allowing these species to produce stuff like the hydrogen peroxide or any microsins? Is it then an environment that's more suitable for that, and maybe that could then point towards
And would it be possible that maybe Citrobacter was already there beforehand and then, E. coli comes in, being, well, not able to establish itself? So would another setup of interest perhaps be that you first grow Citrobacter in a certain medium, use the spent medium to see if that, would that be of any use? Or is that not the situation that we have in the gut?
Yeah, I think I can't say at the moment what the order, the chronology of which one comes in first or not.
Right.
That's just a limitation of the study, and I do think all of the different combinations of having Citrobacter first and then putting E. coli in afterwards, having E. coli first, then putting Citrobacter in, does it manage to kick it out? All of these would be interesting to explore, to really try and pinpoint what the reason is, why it's potentially beneficial, and whether that's either just the species that are there, or grow Citrobacter under stress and take the supernatant, for example, as it metabolites that's produced, and then add that to E. coli. Does it then reduce the survival of E. coli? I definitely think that would be ways to explore it.
So lots to explore.
Yeah.
But we don't have any more time, so I'm giving the word back to the pro-rector, and thank you for your answers.
The opposition will be continued by an additional opponent online, Professor Hall, and she is Professor of Microbiome Research. Professor Hall.
Thank you, and thank you, candidate, and thank you, Matthew, for a very well presented presentation at the start of your defence, and also I really enjoyed reading your thesis as well. So maybe unsurprisingly, I'm going to ask some questions about the microbiome, Matthew. And I think bringing up some of the points that you actually already presented to the audience at the start of today's defence, which is thinking about some of the factors that influence the microbiome. So not just infection with E. coli, but maybe if you could give us a bit more of a broader overview about some of the key factors that are known to significantly change or perturb the microbiome. And I've just got some follow-up questions after that.
Hi, esteemed opponent. Thank you for your kind words and your nice question. Yeah, for definite. I think there's two. I would separate this into two broader categories of the neonatal development of the gut microbiome and the long-lasting effects that this can then have for your adult microbiome, and then obviously adult associated and what's the word I'm looking for? Behavior associated changes that may occur to the microbiome. So starting with the neonatal, I know there's a study that looked at the development of the gut microbiome of neonates based on whether they had vaginal birth or cesarean, and then using stuff like formula versus breast milk. And that is a very significant factor. Those two factors are very, very significantly associated with stuff like the development of atopic dermatitis during life. I think enteric colitis. So vaginal birth, then being exposed to bacteria like bifidobacterium, which are very beneficial to the gut microbiome, and big short-chain fatty acid producers. And then also within breast milk with human milk oligosaccharides, which then have very healthy anti-inflammatory and pro-species effects within the gut microbiome, do have long-lasting effects into your adult life. But then from an adult perspective, I then think it's really a lot more about the behaviours of adults. So whether you're taking antibiotics, although I did see a study that tracked the gut microbiome pre-antibiotic usage and then post, and then continued to follow up, and did see that the gut microbiome straight after antibiotic usage is obviously significantly perturbed, whereas over time it does end up trying to resemble the pre-antibiotic use microbiome, although slightly different. So it does try to bounce back. I would say that those, for example, are two of the main factors.
No, that's great. Thank you very much, Matthew. So kind of following up from that, so obviously you've looked at travellers, and I would assume that they're a relatively diverse bunch, and you talked about behaviours. So one of the questions I would like to ask you, in terms of, you didn't see a difference in diversity in terms of for carriers and not for carriers, but did you also think about these other factors that influence the microbiome, like age and diet, and were you able to stratify based on those additional factors, or impute them into confounder analysis? Or if you weren't able to do that, how would you go about potentially pulling that data apart and being a little bit more nuanced with your analysis?
So I know from the combat study, which is the precursor to my project, apart from the factors I've already mentioned that affect the gut microbiome, like diarrhea and use of antibiotics, I don't think there was anything that really stood out, apart from with stuff like age and sex. It was more the consumption of pork products and food from street vendors, so a very behaviour-related thing, was then associated to picking up disease of E. coli. Granted, levels of hygiene levels, and pork being a potentially easy source of contamination for disease of E. coli. Within my analysis, so at a diversity level, trying to associate this with the acquisition of ESBL coli or not, I did have this data, and when I ran a multilinear regression analysis, incorporated all of these factors of age, sex, BMI within this analysis as confounders, and then reran this analysis based on the region, the sub-region, so North East Africa, South and Southeast Asia, as I thought that was not suitable to include as a confounder because it's such a confounding factor that it's just easier to separate this out amongst them.
No, that's perfect. And obviously, you were looking at this for people that were traveling to these countries. In terms of thinking about maybe other studies that have been done, thinking about people that live in those countries and carriage of E. coli, but other just multi-drug resistant pathogens, how important do you think diet, for example, is with respect to that potential resistance? Or as you say, they might be carrying it but aren't symptomatic, maybe due to the wider diversity of the microbiome, and how diet might be influencing that, Matthew?
Yeah, for definite. I've read at some point that in countries where the consumption of quite volatile foods, so very spicy foods, for example, may actually be a component that's then potentially protecting people because it's with the causes it might have on the gut microbiome. I do think a reason why these strains are at such a high level of carriage is probably partly down to stuff like their diet, and that's why it's maintained. So even though was it half? Yeah, just nearly half of people out of all of our travellers brought this back to the Netherlands. The carriage or the rate of clearance is very, very high. So most people clear this within a month, and then there's a subset of people, maybe depending on which species of E. coli are the ones that are these carriers, or if the plasmid mobilizes within the gut microbiome, there's a very small subset of people that do have long-term colonisation. So I do think a big factor for that is the diet changes when you come back. If you come back to the Netherlands, you then follow a traditional Dutch diet, more likely. And maybe then why we're then seeing a lower rate of carriage or high rate of clearance, but it's not cleared in other countries where the diet is completely different.
Yeah, and how do you think, just final quick question would be, in terms of diet is really important, so how do you think you could kind of capture that more specifically, looking at the diet and how that could be influencing, and more like the actual individual components of the diet? Is there any way that you think that you could do that if you could do it again, Matthew, and redesign the study?
Yeah, I think the way that I would incorporate that would be to track the overall, I can't remember the terminology for them, like the larger components of how much fiber, how much carbohydrates, how much protein, be able to track the consumption that they may have pre-travel, post-travel, or during travel, and then post-travel, and maybe then that will pull out some, these components. Some of these components will then come out to be protective or not. So I know, for example, like with dietary fibers, that's then the precursor for short-chain fatty acids, for example. So a high fibrous diet will then elicit beneficial effects. And that might then come out if you can track these statistics during travel. And then compare
Perfect,
with
that's great.
HDLs.
Thank you. Thank you very much, Matthew, and that concludes my questions.
And thank you that you were present in this degree committee from Birmingham. The opposition will be continued by Professor Willems. He is Professor in Population Genetics of Antibiotic Resistance and is from the UMC Utrecht.
Thank you.
Professor Willems.
Dear candidate, I also want to congratulate you with your thesis, and also want to extend my congratulations, of course, to your comprehensive supervising team. It must be a privilege being supervised by so many excellent researchers, scientists, but I guess it might also sometimes be very challenging with so many supervisors. I would like to, that was not a question.
No comment.
No comment. I would like to start off with discussing chapter 3 again about the competition essays. And my first question is about the choice of strains. You already elaborated on the Citrobacter part, but then looking at the E. coli strain that you use an ESBL-producing E. coli with a chromosomally inserted CXM gene. And that's not typical for an ESBL E. coli, I would say. Normally, these ESBL genes reside on plasmids. So you say something about that in your discussion, but can you please explain me why you choose that particular strain, referring to the chromosomally insertion?
Yeah. Highly esteemed opponent, thank you for your kind words and good question. At this point within my PhD was quite affected by the coronavirus and the inaccessibility of the ideal strains that I wanted. The samples, like from the traveler from the study that I worked on, for example, which then did have all of the strains that I definitely wanted. So to counteract this, within Birmingham, and what I had accessible was a study for people that went to Laos. So I was like, okay, Southeast Asia, place of high concern, and that was also tracking stuff like Enterobacteriaceae. Within this, I then made the decision to select the chromosomally inserted CTXM genes, or the E. coli with those genes, because I didn't want to have trying to have the persistence of the plasmids, and whether that needs to be grown on, if I need to adapt the plasmid or grow it on a antibiotic-containing plate, to then also be able to count, and that will then affect the other species that don't have this plasmid. I didn't want to incorporate that because I thought it was going to be too tricky with the amount of time I had left. So the second best thing I thought was plenty of these samples, well, not plenty, like a low number, but enough of these samples did have chromosomally inserted ESBL genes that it's the second best I can do. And if Citrobacter could outcompete E. coli, then that would already, even like non-ESBLs, might point towards maybe why it's being protective.
Well, now I understand it much better, because in your discussion you talk about confounders with a plasmid containing CTXM genes, but I thought, yeah, well, that is the real situation actually. Yeah. Okay, and then you included, apart from the Citrobacters and your ESBL E. coli, you include a third set of isolates, which were commensal E. coli from healthy volunteers. Mm-hmm. And so why did you want to include that third group? What was the reason to not stick to the Citrobacter and the ESBL E. coli?
For the healthy people,
yeah.
For the healthy E. coli? It was so I can determine whether, granted, even though it was chromosome inserted, whether there was then a difference between commensal E. coli versus the ESBL producers. So I did always try to match based on phyla groups, so I had the same phyla groups within these two populations. And if that would have been a factor that could have been involved, maybe there's something about these commensal E. coli that's then different from the ESBL producing, because we know that they were also picked up during travel. So it's really kind of two groups, commensals versus ESBLs, but also commensals versus travel associated.
Okay, because in fact, I think, so you discussed with your first opponent the fact that the prevalence rates of ESBL E. coli also in the Netherlands, but in Belgium is very high. I don't think that, at least that's my opinion, maybe you have a different opinion, that these ESBL E. coli are basically not fundamentally different from indigenous E. coli, other E. coli that we carry in our gut, or do you think they are? Apart from the fact that they produce an ESBL.
Yeah. I think obviously just having the plasmid you literally could have the identical genomes and then just have a plasmid in one of them and not in the other.
Exactly.
It's why I really try to focus on commensals from healthy volunteers versus the travel associated, because then that's more likely to be E. coli that's difference between Birmingham people versus E. coli picked up from Laos. So I did try to incorporate this within my analysis, but yeah, I do agree that there's not much that could necessarily be different between commensal E. coli and the invading E. coli. It's more if it's pathogenic E. coli versus an E. coli that's just adapted to just living in the gut happily.
Did you at some point thought of also doing the competition between, let's say, your commensal and your ESBL E. coli? because it's not in the thesis.
So it's not in the thesis because I couldn't discover a way to be able to determine the difference between them on a plate easily. I'd be able to count the difference consistent with the other comparisons I was making. But yeah, my initial plan was to do like a qPCR between these different groups and then have these different PCR targets between Citrobacter and the two groups of E. coli's, but then also between the two E. coli's. And if they can outcompete each other, if there was something there that would then answer these questions of are the ESBL producing E. coli different from the commensal E. coli? So yeah, I would have done that with qPCR, considering time.
Yeah. Is there time for another question? I'm asking. Not really. Okay. Then I thank you very much for your answers and give the word back to the pro-rector.
Thank you very much. And I hope you understand that I would give the external host more time. The opposition will be continued by an additional opponent, Dr. Kamench-Chikova. She is an assistant professor at the Department of Health, Ethics and Society in Maastricht University.
Thank you very much. Dear candidate, congratulations on your well-researched and well-written thesis, and congratulations to your team on the wonderful job. I'm not a microbiologist, I'm a sociologist, so I would like to invite you to reflect a bit broad on the implications and applications of your thesis. And for that, I would like to ask maybe one of your paronyms to read proposition 7. Maybe one of the paronyms can do that? Yeah.
As scientists, care needs to be taken to not stigmatize the gut microbiome-mediated transmission of AMR bacteria from low and middle-income countries to high-income countries.
Thank you very much. I was very pleased to see this proposition and was also quite triggered by that because, as you probably know, several years ago, scientific community have engaged in this exact discussion regarding the naming of New Delhi metallo-beta-lactamase. So having already a couple of years since that discussion and conducting your research in a very similar settings, what do you think are the lessons that we as scientific community should learn in order to avoid those type of stigmatizations? What would you advise to your fellow scientists?
Hi, esteemed opponent. Thank you for your kind words and good question. It's a tricky one because obviously it has to be done with so much nuance and care, and because it's such a, like for me it would trigger such an emotional response if someone was just like, we don't let foreigners in, for example. It's so easy to just really want to counter that really aggressively. I think some lessons really is there's such a high level of globalization regardless, from both rich people going abroad on holidays, for privileged people going abroad on holidays, as well as people like refugees that may be leaving countries where there's endemic rates of carriage of ESBL coli or other species that are being forced to move countries. So there would be, for me, there would be a bit of hypocriticism for people that would try to stigmatize people like refugees, but then not even consider people like international travelers. Both are kind of mandatory, I think, both the movement of refugees and the fact that a lot of the different countries' economies rely on stuff like the tourism industry. I don't think we can really put rules on anyone. It's more how we then change the outcomes of maybe increased levels of, for example, ESBL E. coli coming into our country, and then how we might tackle that, rather than the people. It's just something we have to work with rather than work against, if that makes sense.
Yeah, it does. Thank you. I think it also challenging. Of course, we know from COVID that data from scientists have been used to also stop traveling from some countries, but not from others. So maybe it will refer back to the first question was asked by Professor Kubov, what kind of actions you would recommend to public health bodies, to policymakers, informed through your research that we should put in place?
Yeah. I think then the avenues that I would like to more speculate with would be, can we pump more money into producing stuff like vaccines, maybe against stuff like ESBL E. coli, and really try to use the data towards changing how much money and how much focus goes on to preventing the species of, and the problem with the AMR species, rather than the people. So I think that's, I would say like production of vaccines would be where I think this would be best incorporated.
Well, thank you very much. I'm actually really happy you mentioned the vaccines, because I'm not sure if I have time, but I have a very quick second question. Because indeed, in the conclusion of your thesis, you mentioned that travelers' diarrhea is the important component for travelers to acquire antimicrobial resistance. And you mentioned that it is important to focus on the prevention of travelers' diarrhea. But reading this and agreeing with you along the way, I was again quite puzzled by your proposition 8. Maybe your other paranym can read it out.
Shall we skip this part?
Oh, yeah.
Because,
okay, there is no time. But I'll just quickly summarize it. In that proposition, you mentioned that targeted intervention strategies for minimizing the transmission of IMR should focus on clearance of bacteria in susceptible people rather than prophylactic interventions. Wouldn't that go in contradictions to your recommendation for diarrheal prevention?
It does, yeah. When I wrote that, it's my opinion on this was kind of changing the more time went on, really. Realistically, I think both are in place. If that was removed, rather than prophylactic interventions, would make this a lot more simple. I think there's just two avenues, really. Of travelers' diarrhea is something we want to minimize anyway, regardless, just because that's just better for people. But also because it was associated with the ESBL acquisition, then that was a really big target. Thank you.
Thank you. Dear candidate, Matthew Davies, the time appointed for defending your thesis has passed, as you noticed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request that you and your company await the results of our deliberations and our return in this room.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Outside. Yeah, that's fine.
7 miles in my rearview mirror. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Traveling so far from gathering knowledge. I put the time in to get everything each of them go And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Outside, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and night. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and night.
Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Oh, thanks. Appreciate it. Literally couldn't have gone any more worse.
Matthew Davies, the degree committee here present has assessed the quality of your thesis and of your defense and of your beautiful presentation. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of Doctor. Professor Penders is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present today, I hereby confer upon you, Matthew Anthony Davies, the degree of Doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the Rector, the Secretary and the other members of the committee and affixed with the official seal of the university. Dear Dr. Davies, dear Matt, congratulations. After having already passed your Viva in Birmingham, you have now also accomplished the very final pièce de résistance here in Maastricht. You can be very proud of yourself. And of course, I would also like to extend my congratulations to your parents and to Alex. And as we've heard all today, your PhD thesis is all about bacteria picked up by international travelers, illustrating that these bacteria do not care about national borders. And by comparison, your PhD was also an international one, moving back and forth between the UK and the Netherlands. But in contrast, you had to struggle with some newly formed borders, newly barriers and restrictions at some points. It all started in the fall of 2018. You applied for a joint PhD position between University of Birmingham and Maastricht University, and the project funded by both universities was conceived by Willem van Schaik, Alan McNally, the two supervisors in Birmingham, and together with Petra and me. And from the very start, you were very enthusiastic about the topic of the project, but you were a little bit more skeptic about having to deal with 4 supervisors, as we already heard before. And I think you were afraid that we would never come to an agreement between the 4 of us. But I guess over time we have proved you wrong, because we were very much on the same page. And therefore I would also like to thank Willem, who could unfortunately not be here today with us, and Alan for this fruitful collaboration. And I also hope that we can continue to collaborate when you have finalized now your PhD. Your PhD journey really started at rocket speed. Already within the first weeks, you visited Maastricht to attend our annual research day and met all of your Maastricht colleagues. And in the meantime, you also made sure that you gained access to all of the metagenomics data generated by the Dantas lab in the United States. After having spent a little bit less than a year in Birmingham, you started your period here in Maastricht, and you soon felt at home. and so much that in the end you didn't want to return back to Birmingham to finish your PhD. And I'm still not sure why. Was it the krentenbollen? Or was it the fact that in the meantime you have met Alex? You can tell us later. One way or the other, in Maastricht you started with the statistical modeling of the metagenomics data, a research field that was quite new to you. Longitudinal data characterized by its sparseness, zero-inflatedness, and compositional nature. This was quite a tough concept to wrap your head around, but you managed, and were in the end the one that could best explain the concept of zero-inflated beta regression model with random effects. And additionally, you also set up the metagenomic species assembly, using the canopy clustering approach with the assistance of the developers in Copenhagen. And this generated some very interesting results, and at your very first conference, the Gut Day in Amsterdam, you were selected as the runner-up for the best presentation. And I think today we have seen why that was the case, because also today your presentation was brilliant. And this is quite a big accomplishment so early in your PhD. So this might all appear as if everything was going to be a straight road to success. But in the meantime, Brexit was causing a lot of uncertainty, and that was also something that was heavily weighing on you. And just a few weeks after this Gut Day, there was suddenly a pandemic, followed by lockdowns and closure of lab facilities. So this also meant that we had completely to shift our focus, because future work on culturing the E. coli strains for analyzing strain level diversity as well as plasmid profiling could not be executed anymore, and we needed to switch to in silico analysis. And during that time, the progress meetings with Petra and me were often more like motivational speeches, trying to convince you that a PhD is also having to deal with these setbacks. And I think you did well. You grew as a person and as a scientist. And in the end, the results were published in a respective journal, Gut Microbes. And by that time, it was already time to move back to Birmingham. And that's also why I want to give now the word to Alan, who also wants to say a few words on behalf of the other part of your supervision team in the UK.
Thank you.
Thank you, John. And congratulations again, Matt. And congratulations, I understand your parents are there today, so congratulations to them, and Alex is there also. I'm pleased they're there to share it with you today. I'd also like to say a massive congratulations to John and Petra, and a huge thanks to John and Petra. I know they were enormously supportive of you during your time in Maastricht, Matt. And of course, as John's alluded to, what started off as a project that should have been joint amount of time between Maastricht and Birmingham, you were caught with the terrible luck that as you were in Maastricht, the COVID pandemic started, and rather than being in the wonderful post-industrial heartland of the UK that is Birmingham, you were in the beautiful Roman time, ancient, beautiful city of Maastricht, and you're probably one of the few people in the world that was quite happy, I imagine, that there was a travel restriction when COVID happened, and that you could spend more time in Maastricht. I think I echo the statements that have been made about your presentations, Matt. During your PhD, it's something that you've absolutely excelled in. You made a presentation to a microbiome conference in Birmingham, actually, that was hosted by the Microbiology Society. And everyone at that commented on how beautiful your presentation was and your presentation of data. And it's been great to see you develop as a scientist and develop as someone who's so good at presenting your data. I would just like to remind you, to pick up on John's point, that you published a wonderful paper, that we do still have a metagenome assembled genomes paper to publish, and I look forward to seeing a very near final draft of that in the very near future, Matt. Thank you very much. It's been a joy to work with you. I'd just like to add, I've WhatsApp messaged Willem about 5 minutes ago. He would like to send his heartfelt congratulations. He's very, very sorry he can't be with you today, Matt. I'm also very sorry I can't be in Maastricht, which was the original plan. Well done again on your achievement, and thank you very much for all your hard work over the last 3 years, Matt. Thank you.
Appreciate it.
Very good. It's good that they can hear you, so that's great. Very good. So, dear Dr. Davies, also on behalf of Maastricht University, I congratulate you with your second degree, which has been accumulated on one certificate. As you noticed, the University of Birmingham is on your certificate now. And I understood that instead of the 3/4 of an hour that you were here in Maastricht, our guests in your position, you had a 3-hour defense in Birmingham, like it's
3.5.
3.5 hours. And you did, but you did pass the test. I understood. Luckily, from the laudation, I had some more information about your last proposition. So my questions on that point are answered now. What we're going to do is make a picture with you and the paronyms and the committee and the online opponents very big on the screen. So it's a challenge for the photographers to capture all this. Followed by a reception function in a room called the Refter, which is the former mensa or dining hall of the congregation that lived in this cloister way back. But please follow us, also you follow us, and then you will get there. But first, the photo, and with this I close this session, and when we have made the photo, you can, through your microphone, also talk to Professor Hall and Professor, the professor, I cannot repeat her name.
Yeah.
You know the pronunciation.
Professor Gagan.
Gagan. Okay. And I close this academic session.
Thank you. Oh, to the front? Okay.
Do you also get the online uploads for the screen and all the people?